A very good evening to everyone. Let us start with the topic of now, that is the spontaneity of mutation. So, as we say in the starting, let's crack CSIR-UGC NET together. I'm your educator Farida Bandookwala, and I'm pursuing my PhD from School of Biotechnology DABD. I've done my MPhil in biotechnology, and I've qualified MP SLET examination 2008, and I have a 10 years of experience in the field of life sciences. Before beginning with the topic of today, let us have a small introduction about Unacademy. Unacademy is one of the India's largest platform for the preparation of CSIR-UGC NET. And if you are opting Unacademy for your preparation, you are doing one of the best thing you can do in today's time, as you can get live classes. The whole course is very structured because there are 13 units. It's very difficult for a single person to attempt all the units together, so the whole structure is quite structured. Whole thing is quite structured, and there are live tests and quizzes, and with the help of live tests and quizzes you can easily evaluate how much you have learned so far. And there is unlimited access for all the videos are you whichever you are opting for. Like if you are going for CSIR plus platform, so you'll be getting all the videos in that category. And with Unacademy, the best part is learn anytime, anywhere. Learn anytime, anywhere. If you're not in the town, if you're somewhere out vacationing, then you can attend the lectures live on your cell phone, on your laptop, on your iPad, whatever. So that is the best thing on Academy. You can learn anytime, anywhere. Then Unacademy is providing lots of free live classes, and these free live classes are on YouTube, and there are special classes. So you for YouTube class, like you have come for come now, so the same thing for YouTube classes. And for special classes you need to download the app, that is Unacademy learning app. There you can make your account, and you can opt for CSIR UGC NET Life Science, and you'll have the access towards all the special classes which are done in this special category. And these classes are also free of cost. You don't have to pay anything. Other than this, there are plus subscription. And what about this plus subscription is that you need to pay a certain amount, but that will be like quite pocket friendly, and you don't have to spend lots, and you will get the whole thing at the fingertips. So what I would suggest is, if you are a MSc, go for a yearly program, that is going to cost you around 15,000, and use my referral code FB01 for getting a 10% discount. So this is how you can get this subscription. And after that, we'll begin with the topic of today, that is spontaneity of mutation. So we are talking about mutation in terms of evolution, that how mutation contributing towards variation, and how mutations are giving rise to evolution. So what are the actual factors which are contributing to phenotypic variation? Like we all are similar, but still some are too tall, some are too short, or some are like average. And whenever a new generation is approaching, we see that the height is like keeping on increasing and increasing. The average height is increasing and increasing. So what are these factors which actually contribute in change in the phenotype? That is the topic of today. So where does the variation come from in the very first place? So we know that each and every individual, all the life forms, they have originated from a single being. There were no many individuals, no different life forms originated at different places, but they all have originated from the very same place. So where does the variation come from in the very first place? Where does this variation come from? So the environment, it plays a major significant role in pattern of variation among the individuals. The very first thing is environment. Environment is the reason for bringing any kind of change. If there is need of change, that need of change is posed because of environment. The ultimate source of variation, that is the fuel for evolutionary change, is mutation. So variation is the thing that is giving rise to variations, are fuel, that is mutation. So mutation will bring lots of variation in the genome of an organism, which will be actually exhibited in the form of phenotype. And these mutations, they are actually because of what? Environment. Environment is the one which is giving rise to mutation. So this is how at the very first place, how the individuals that are becoming different from each other. So environment is the leading cause of mutation, and mutations are giving rise to variation. Then the types of mutation: point mutation. If a single nucleotide is changed in a genome, it is called as point mutation. If there is extra addition of nucleotide, then it is insertion. There is something extra. If there is something eliminated out, that is deletion. Now something is lesser in the whole genome. Then gene duplication. There will be duplication of the original gene which was there. It is going to be duplicated. It is going to be present in one more copy. That is gene duplication. And there is inversion. Now previously the sequence of the gene was a particular kind of sequence, like over here you can see this violet, blue, then this beige, and then orange, and then green. But now after inversion the sequences just change: green, then orange, and then this beige, then blue, and then violet. So the whole sequence is inverted. So this is going to have a profound effect on the expression. So that is how the variation would occur. Then chromosome fusion. There will be 2-3 chromosomes which will fuse with each other, and it will give rise to chromosome fusion. The chromosomes might fuse with each other and give rise to a very different type of chromosome. And then gene duplication, genome duplication. The genome duplication is like making the whole set only in one more copy. Like if there was a single copy, haploid copy, it is changing into diploid. Diploid is going to change into quadruplet, and like this it is going to just increase in number. So gene duplication, there are various plant species which are not actually diploid, triploid, or haploid like that, but they are hexaploid, heptaploid, octaploid. So this type of ploidies also exists in plant. So major source of the genetic variation, genetic change is the mutation, which is the fuel for evolution. Without mutation, there would be no evolution. So the neo-Darwinism is also actually saying that mutation and natural selection. So in neo-Darwinism is kind of a amalgamation of mutation theory that is given by Hugo de Veris and Darwin. So this is the fuel for evolution. So point mutation, what is going to happen? Base pair substitutions in DNA sequences, and what is the cause? Chains errors during the DNA synthesis or during repair of DNA. And what is the significance of such kind of point mutation? It will create new alleles. Then chromosome inversion. So what is going to be its effect? Alleles inside the inversion are locked together into a unit. Alleles inside the inversions are locked together into a unit. And then gene duplication, the extra gene is free to mutate and perhaps gain new functions. So if already normal copy is present and it is going to make one more copy, so now you can edit it a number of times. Like if you are having a word file in your computer and that is your original file, and you just want to make some changes and see whether it is working out or not, you won't go on making the changes in your original copy. You're going to just copy that thing and paste it somewhere, rename it, and then you start editing it. So that is how gene duplication is there. So we have got a functional copy, fine, no worries, it is carrying on life very well. But now when a different copy has come, it is creating some new changes. So it is a very good location for accumulation of any kind of changes. Then polyploidy, it can create new species. So instead of haploid, diploid, it is going to create ploidies, lots of ploidies. And because of this, there will be generation of lot many species. The phenotypic effect of mutations depends on the location of mutation. Now, any kind of mutation, it depends on what? It depends on the location of mutation, that where it is actually happening. It is happening in the heterochromatin area, it is happening in the euchromatin area. All these things are going to matter so much that the place of mutation makes the major difference. So phenotypic effect, whatever is being exhibited in the organism depends on where it is happening actually. So that is also one thing. What is happening is also important, and where is it happening is also important, and what is it is going to do is also important. So all the things are important. You cannot skip one of the things. So if it is happening in heterochromatin area, fine, no worries. It's not going to give you a direct impact, but it is going to accumulate years after years, millions of generations, and then suddenly something new will come up. So that is like the mutation in the heterochromatin area. That is the significance of heterochromatin area. It's not going to give you immediate effect, but if it is happening in euchromatin area, yes, it is going to give you immediate effect, that it happened now. Okay, fine, this is the final consequence. So location of mutation, types of mutation, and consequence of gene action. So coding region, what happens in the coding region? If it is substitution, insertion, deletion, or duplication, it alters the product of the gene and thus its function or activity. And cis regulatory regions are the substitution, insertion, deletion, duplication that are going to that alters the binding affinity of promoters, activators, repressors, etc. And it is also going to give rise to, it alters the timing, location of the level of expression. So when it is going to be active, when it is going to be inactive, that all thing is going to be disturbed because of such kind of things, and it alters the developmental environment context in which the gene is expressed. It will change the whole environment that when it is happening. In transregulatory region, mutation to coding regions of the transacting factors alters the binding affinity and thus the activity of promoter, activator, repressor. Alters the binding affinity and thus the activity of a promoter, activator, and repressor. All of these things are going to be changed because of the trans-regulatory region. So trans-regulatory regions, they are not directly affecting. It is going to change the untranslated regions, upstream regions, and downstream regions. So these are that kind of things. And this regulatory region, they are going to change the timing. And trans-regulatory region is going to change the affinity, their combination, these all things it is going to change. Then physiological pathways, example hormones, it alters the timing, location, and level of expression of the gene. It alters the developmental or environmental context in which the gene is expressed. So this is all you can see from the phenotypic effect of mutation. Then point mutation, it seems that it is just a single nucleotide. What is it going to do? Is it going to bring lots of difference, or no, it is going to bring minor differences? So all these pictures what I have included over here is actually because of the result of point mutation. So you can see that so much of problems can arise because of a single point mutation. See, this is a whole albinic family here, here, and here, and these two, they are like normal. So these are because of the point mutation. And is this a normal trait to have the joint fingers? No, we are supposed to have free fingers. So this is also because of the change in a single nucleotide, which is going to change the amino acid sequence and which is going to stop that function, or it is going to function in a different way. Here in vitiligo, here difference in the backbone structure, then macrocephaly is going to happen because of the point mutations. Changes in the nucleotide sequence can alter the amino acid sequence and change in the protein structure, and it is going to introduce the stop codons in many of the cases, and that is going to give rise to lots of problems. So point mutation in regulatory regions can alter the timing level of the gene expression. This is what I wanted to tell. Then another thing which is responsible for mutation and which is in the end responsible for evolution is transposable elements. So transposable elements, they are also called as jumping genes, and they were discovered by Barbara McClintock in year 1983. So these jumping genes, they are also responsible for introducing any kind of new variation in the whole genome of an organism. So that is also one of the factors responsible for the changes. And here is some of the example of the jumping gene, that whether they'll be having jumping genes or not. Now, next is chromosomal inversions and chromosomal fusions. So, chromosomal inversions, the whole thing will be just inverted out. It is going to get inverted. See over here, it is going to get inverted. And if it is going to get inverted, it is going to have different effect than pericentric inversion, which is more devastating because it is occurring around a certain axis or around a certain point. It is not like in the similar region. Here it's in the similar region, but here it's just changing the axis, so it is going to pose more problems. Then chromosomal fusions, two of the chromosomes, because of some unfortunate conditions, they might join together. Chromosomal fusion. Gene duplication is a major problem in many of the things. So in this table, the number of duplicate gene column refers to the number of loci in the various gene families. These loci are presumed to be a result of duplication events, and they have high sequence homology, code for products with the closely related functions, and are often clustered close to one another on the same chromosome. They are on the same chromosome and they are clustered together very closely. The name of the family and name of the duplicated genes here it is written. So loci found in many organisms. Actins are present in 5 to 30 copy. Tubulins are present. Myosin, keratin, histones, heat shock proteins, then in insects, eggshell protein, then in vertebrates, globulins, globins, alpha like globins, beta like globins, all these things they are in a very duplicated form. So maybe some kind of change is being accumulated in one of the gene and it is not showing us now because there are so many other genes to take over the whole process, and smoothly it is taking the whole course in a further direction. So that is the gene duplication. Then the fate of duplicated genes, we are going to talk, retain their original function and provide an additional copy of the parent locus. And now this additional copy is going to be very notorious and it is going to accumulate the changes, accumulate the point mutation and become functionless pseudogene. So like if they are duplicating, it is not that it is always going to be hyperactive and going to create difference and difference. Yes, it is going to create difference. No, they can become pseudogene also, they can become silent also, and they can just remain like this forever. And they may accumulate some kind of changes which may give rise to some kind of variation and speciation. Gain a new function through mutation and selection. Yeah, that is also one of the things. New functionalization can also occur. While humans and chimpanzees are 1 to 2% different at the nucleotide level, they're more than 3% different in the gene copy number. So we say that the chimpanzees and humans, they are just 1 to 2% different. There is no much difference in humans and chimpanzee in the whole genome. So we have just seen the genes. Okay, genes, whole number of genes, the whole 46 chromosome. Okay, fine, they are 1 to 2% different, but the major place where they are differing is in the number of the gene copy. So now are you understanding the importance of copy number of the gene? So if the copy number is also going to change, it is going to create some kind of difference. So within human population, many disease phenotypes are linked to copy number variants. So we have already seen some of the examples which are related to point mutation. There are many things which are related to problems in the humans which are really disease linked. So different classes of mutation occur at different rate in humans and affect different proportions of the genome. So in this y-axis we have written number of mutation, and x-axis we have written various kind of mutations. So number of mutations, and then point mutations, small insertions and deletions, insertions large, duplications and deletions, and gain or loss of chromosomes. So this is the number of mutation, and here total base pairs of mutations contributed by each category. So the category which is contributing highest in the change in the gene number is, first of all, okay, if there will be gain of chromosome, whole chromosome is coming, and if there is loss of chromosome, whole genes on that chromosomes are going away. So definitely gain or loss of chromosome is one thing, and large duplications and deletions is standing second, though this is standing second, but it is posing a very important effect. It's not at all okay, but it is, yes, it is impacting a lot over here. because it is going to give large number of base pair changes. If point mutations, if there are 100 point mutations, and 100 point mutations are going to give rise to 100 differences in that. And if there is duplication, all of a sudden there will be 200, 300, 400 changes. So this is posing a great change in the whole genome. So large duplications and deletions, they are going to create lots of change. So in exam also you have to take that gain or loss of chromosome is first number, then large duplications and deletions second number, point mutation fourth, small insertion fourth, insertion of mobile element is contributing quite lesser in this category. So do not forget this table at all. The genomic mutations appears to be roughly constant in haploid microbes and simple eukaryotes. So genomic mutation constant in haploid microbes and in the simple eukaryote. So how is it that organism we have seen written over here? Page M13, page lambda, page T2 and page T4, E. coli, yeast and neurospora, and genome size you can see from here per base pair like this, and these results suggest that the genome size has increased the DNA replication repair machinery, and it has increased in efficiency. So if the genome size is larger, the mechanism of correction has also increased, because now they have to take care of so many base pairs, so the machinery needs to be improved. Then comes constancy of mutation rate. So is the mutation rate constant, or it is fluctuating like that, or it has to be always constant, that yes, after 1 year these many mutations are going to be there? No, they are not going to be accumulating, is it so? Or it has to be there. So the genomic deleterious mutation rate is 0.00 per cell division in E. coli and 1.5 per generation in Drosophila melanogaster. So that is like it is going to be there. Constancy of mutation. Mutations are constant. Mutations are, yeah, they are random, but they are going to occur in any of the case. So high rates in flies and humans suggest Drake's constancy hypothesis cannot be extended to higher organisms. So mutations are going to accumulate, but exactly not in a calculable things. So there is one hypothesis which says that males are going to accumulate lots of mutations as compared to female. Females are going to accumulate less changes, and males are going to accumulate more changes till a certain age. The mutation rate in males is on the order of 10 times that in females, probably because of higher number of cell division from zygote to gamete. And female cell divisions are 24 independent of the age. Whatever is the age, the egg which is formed is actually come from 24 divisions. And male cell divisions are actually 36 plus whatever is the age minus 13 into 23. This is the constant formula. So whatever, if a male is of 20 years, its sperm has been undergone this cell cycle is 200, and if it is at the age of 45, the number of cell division it has been through is 770. So this is the mutation rate. So mutation rate in the males is on the order of 10 times of that female. So males, they are going to accumulate more of the mutations in them. So the male rate of mutation is approximately 1.2 into 10 raised to power minus 8 per base pair generation, or approximately 1 into 10 raised to power minus 10 per cell division. The genetic mutation rate is approximately 36, and more than 6% of newly fertilized eggs carry a gross chromosomal abnormality. Approximately 5.5 of these terminate as spontaneous abortion. So if a newly fertilized egg, it is carrying some kind of chromosomal abnormality, it is going to get aborted on its own. No one has to make lots of changes in it, but it is going to be aborted on its own. There will be no other survival mechanism for it. So father bequeaths more mutations as they age. So genome study may explain link between parental age and conditions such as autism. If a father is of higher age at the time of childbearing, it can have a problem related to autism in the child. And for mothers it is not that much. So in 1930, the pioneering scientist JBS Halden noticed a peculiar inheritance pattern in families with long histories of hemophilia. The faulty mutation responsible for blood clotting disorder tend to arise on the X chromosome that father passed on to their daughters, rather than on the mothers that passed down to their daughters. So Halden subsequently proposed that children inherit more mutations from their fathers than their mothers, although he acknowledged that it is difficult to see how this could be proved or disproved for many years to come. So there is no concrete data to actually affirm this concept, but it is seen that father with older age have more of defective genomic genome in their offspring, because they have to undergo lot many divisions as compared to the mother. The rate of de novo mutation and importance of father's age to disease risk was actually published in a research paper that was like in the year 2012. So what does it say? That after accounting for random poison variants, father's age is estimated to explain nearly all of the remaining variations in the de novo mutation count. So this is like a paper which actually supports the data which was given by Halden, that what Halden was saying. These observations shed light on the importance of the father's age on the risk of disease such as schizophrenia and autism. Then directed mutation. Do mutation arise spontaneously or in response to selective challenge? So this is the picture over here. In the pattern A, the mutation is directed, and in the pattern B, the mutation is altogether very random. It is not at all directed. It is just if it is occurring in one thing, okay, it is keeping on occurring and occurring, and in one it is directed. So it is seen that mutation is actually are mutation random. So for this, the accumulation of, there was another experiment which was done on lag system, and there it was um, seen that the mutation is actually occurring in a sequential manner, in a very distributed manner in the whole series of experiments. So mutation rates after 10,000 generation of evolution of ancestral population, high mutation lines are there. There are some genomic sequences which are more prone to mutation, which are giving rise to mutation in a very constant manner. You can see from this chart that how it is happening. Then what is going on now for establishing the fact is recent studies by Rosenberg and Foster suggest that alteration of the recombination repair pathway is essential for this result. Starvation is mutagenic either as an unavoidable consequence of physiological deterioration or increasing the mutation rate, but adaptive in the sense that not mutating is certain death. These mutated strains may have a short-term advantage coping with environmental stress, but over the long term, they will be at a selective disadvantage. So people going in long-term starvation can be at a lower pedestal as compared to people who are having a very systematic routine of diet. Then mutational effects. Fisher argued that mutation of a small effect are more likely the fuel for evolution by natural selection. So the smaller mutations which are very hidden, they will keep on be in the secret form or hidden form for a long generation. But suddenly they can come up. So they can be silent for a long duration, but they can come up with a massive effect. So small effects are more likely to be there. Then mutation accumulation experiments, how are all these things going to happen? Similar plots to figure 12.3 or for 50 lines of bacterium. Here you can see that single bottleneck for 300 days. Here the fitness is measured as the exponential rate. So fitness is going to decrease and decrease as the mutations are going to increase, but after a certain time they will be profound, and they can have a selective or selective advantage or selective disadvantage. So conclusion from mutation accumulation studies: the majority of spontaneous mutations have a slightly deleterious effect on fitness, as I told you that initially it will be deleterious for the fitness, but then it can be actually accepted by the nature. The average effect of spontaneous deleterious mutation is 1 to 2%. decrease in the fitness. So whenever mutation will occur, immediately it won't be accepted. Immediately it won't have a immediate effect of positivity. It might be deleterious. Then summary of knowledge of mutation rate. The spectrum of mutation is enormous. The mutation thing we can talk on and discuss for a long duration, but one has to sum up it somewhere. So ranging from chromosomal rearrangements and duplication to insertion and excision of transposable elements to single base pair substitution, insertions and deletions. The mutation rate is subject to evolutionary modification. Mutation is actually the basic key of all kind of evolution. The vast majority of mutations appear to be deleterious. So mutations, they can be deleterious. Mildly deleterious mutations are much more common than lethal. So the mutations which are which can be deleted or which are deleted are more common than lethal. Immediately, lethal mutation would not occur. Initially, the mutation would be milder and then it will be then taking up the course. The mutation rate per generation increases with the number of cell division in mammals. The point mutation rate is much higher in male than in females. We have seen this in detail. Then summary. So for polygenetic characters, the mutational rate of introduction of new variation is on the order of 0.1% to 1% of the standing variation. The adaptive value of mutations change with the ecological circumstances. The mutations arise randomly with respect to their utility. The mutation rate can be modified greatly by the environment. Mutations are actually affected moreover by environment. Environment plays the most important role in the mutation. So they can be random, but in the environment, it can give a large impact. So like this, we have completed this topic of spontaneity of mutation. So as we say, let's crack CSIR-UGC NET together. For getting all the notifications of the upcoming video, please hit the bell icon and please like my video if you have gained something or if it was knowledgeable, and use my referral code for getting a 10% discount. And lastly, do not forget to comment in the comment box if you want a particular topic to be read by me, or you want me to explain that topic out of all the 13 units. And if you are having any doubt also in this particular topic, you can just write down in the comment box. It will be like whenever I'll be going through my videos, I'll just check out that what question you are writing, and then we can just discuss that thing. I can comment you over there if question needs to be solved, and whatever query, whatever conversation you want to have with me, you can write in the comment box. So this is it for now. Hope you have understood the concept, and we'll be meeting tomorrow with another topic. And this is it for now. Be happy and keep on studying. It's about